Welcome again to another episode of the Nuclear Medicine, Molecular Medicine podcast, and I'm really privileged this time to have a very interesting speaker with me today, John Seibyl, who's Professor at Yale and Director of MNI. I'll get him to explain a bit more about where he works. Perhaps he can start by doing that, and then we're going to talk a bit about what's changed in amyloid PET imaging since we last did a podcast on this 18 months ago. So.
Good. Well, thanks, Rob. It's good to be here, and I'm happy to talk a little bit about my experience with amyloid imaging and start with a little bit about what I do and where I'm from. I'm the founder and president of a company called Molecular Neuroimaging, which is based in New Haven, Connecticut in the U.S., and also the executive director of a private research foundation called the Institute for Neurodegenerative Disorders, also in New Haven, where we do research in brain imaging and develop radio tracers that interrogate specific targets that are relevant to neurodegenerative disorders like Parkinson's disease and Alzheimer's disease.
Okay. Perhaps we could start, and I just mentioned that John's out here doing a masterclass in Australia, and sent out here by Cyclotech. I think they'd like me to mention that, but they've been very useful in doing that for Australia. Perhaps we can start by looking at what's changed. About 18 months ago, we had the first FDA approval of a tracer, and there were, at that stage, there were some research projects using that for amyloid imaging. But now we've had some of the results come through, and some of the new research projects coming through, and amyloid imaging has changed in terms of what it's useful for, or what it should be used for. Perhaps you could talk a bit about some of the research projects that haven't worked, and some of them
Sure.
that are upcoming.
Sure, fair enough. Well, one thing that's changed over the last 18 months, at least in the U.S., is that there is now approval for 2 amyloid imaging PET agents. And they are approved for aiding in the diagnosis of difficult-to-diagnose patients with suspected Alzheimer's dementia. There is actually a third agent that is expected to be approved sometime perhaps this year or early next year, and that's the agent from Piramal called florbetaben. So now there's in the market, commercially available, 2 and maybe 3 amyloid imaging agents, and the clinical acceptance of this has been hampered in the U.S. by the fact that there hasn't been reimbursements for these studies, and so there's been really limited experience clinically. While all this has been going on, there's been a lot of research in the therapeutic world in Alzheimer's disease, specifically therapeutics that are designed to eliminate or reduce the amount of amyloid protein in the brain, and of course amyloid protein is thought to be really important in the pathophysiology of the disease. It's an accumulation of an abnormal protein that happens very early in the disease course. In fact, it may happen 18, 20, 22 years prior to the manifestation of dementing symptoms. So we've got potentially this marker for a disease that is devastating were you to get it. We have a tool to assess that, the presence or absence of that marker. And now what the studies are really focusing on is identifying people who may be at risk and who may be best benefit from an anti-amyloid therapy. Meanwhile, while all this is going on, there have been any number of phase 2 and phase 3 clinical trials with antibodies and secretase agents, medications that are designed to reduce or eliminate the presence of amyloid in the brain. And up to this date, they've all been failures. That is, in patients with probable AD, there hasn't been shown to be a clinical effect. The interesting thing about these trials that has changed over the last 18 months is now, as these trials are being designed, amyloid PET imaging is being incorporated into the selection criteria, so that for the first time, patients are getting a scan, and if the scan is positive, then they are enrolled and they meet the other criteria, then they're enrolled in the trial. If the scan is negative, then they don't really have the pathology that the treatment is designed to affect, so they get excluded. And what's been interesting is that as many as 20 to 30% of patients who are designated for potential enrollment in these trials end up having a negative scan. So that is a really surprising finding, because it means that were those patients not to have an amyloid PET scan, then they would be inappropriately enrolled in a trial for which there would be no expected benefit. And so now screening has become essentially the essential component of enrollment in these anti-amyloid trials. Amyloid PET imaging is also being used to monitor changes once a patient is enrolled in a trial, specifically using some quantitative measures like standard uptake ratio values and things in regions of the brain that are most associated with amyloid imaging. The patients are scanned serially over a course of sometimes a year and a half, or 2 years, and the effects of the treatment is evaluated against the brain scan to see if there's changes. And while the readouts from the data are just starting to come in now, one thing that we've learned from these studies and from other studies like the ADNI trial is that there's a slow progressive increase in the amount of amyloid in the brain over a period of years, and that at least in one trial there was shown to be, in a sub-study, a reduction of that increase over the course of a year and a half. Now, these were very small sub-studies, and they need to be replicated. So the amyloid PET has really become important in the therapeutic development of anti-amyloid treatments. As I said, the trials have been failures, with the exception of one very interesting trial, which is a drug, an antibody called solanumab, which is an agent that's being developed by Lilly in the U.S. and the rest of the world. And they did a trial in mild and moderate Alzheimer's subjects with probable Alzheimer's disease. And the trial failed. That is, it didn't meet its clinical endpoints. But they did a sub-analysis in patients who had more mild illness. And in those patients with more mild illness, they found that the treatment with the antibody actually slowed down the rate of progression, as measured by a measure called the ADAS-Cog. Slowed it down by about 30% compared to the placebo condition. And so while the trial failed, this small subset of patients with more mild disease showed a treatment effect which was statistically significant. And as a result of that, people became very excited all of a sudden that now there has been a demonstration, albeit in a small subset of a larger study, of a potential disease-modifying influence of an anti-amyloid antibody treatment. So that data was reported about a year ago. And it really has changed the landscape and the energy now that pharmaceutical companies are being devoting to the development of their antibody trials. And so Lilly has already launched a next large phase 3 trial, where instead of focusing on patients who have more moderate symptoms, where the idea may be that maybe a little bit too late, that the disease may have progressed enough, that it's really not the population for whom these therapies might be best intended. They're focusing on more mild patients and enrolling in a trial called the Expedition 3 trial, which is the kind of the next generation of the trial that they ran that showed these initial very promising results. And of course, amyloid PET imaging is again serving a very important role in that trial, a role of both screening and selection for eligibility, as well as monitoring the progression or the potential efficacy as an imaging biomarker over time. So people are now moving to this notion that if we're going to use an anti-amyloid treatment, that the treatment really needs to be focused on earlier patients, not only those with mild AD, but even prodromal patients, or sometimes they're called MCI, mild cognitive impairment patients, and even before that. So there's some interesting trials that are being developed to evaluate these very mild patients. Probably the most I guess challenging and an interesting trial of them all is a new study that is just being started. It's an investigator-sponsored trial called the A4 study. And that study is not looking at patients with a diagnosis of mild probable AD, but it's evaluating patients who are above a certain age who happen to have a positive amyloid PET scan. And if they do, then they're asked to enroll in the trial where they are treated with either placebo or active antibody therapy. And the idea is to follow these subjects. They're not patients because they have an abnormal scan, and to see if that treatment slows down or prevents the onset of cognitive decline, with the idea that if you happen to have a positive amyloid PET scan, then you are likely to develop some cognitive changes within some period of time. And so that trial is interesting in that it's not clear how many subjects need to be screened in order to get those positive patients. And so as a result of that, working with Lilly, who is providing the drug, and the NIH, which is in the U.S., which is providing a lot of the funding, AVID and other consortium members, we've developed a very rigorous screening criteria to really ensure that we pick up potentially very subtle or increases in amyloid deposition in these normal folks who come in off the street without any complaints of cognitive impairment. And so amyloid PET imaging is now being used in some really interesting ways in therapeutics. And I think the interesting thing from my perspective is that there's a lot of translation of this into ultimately clinical practice, where the idea of how do you, in the event that we live in a world where there is now, for the first time, a disease-modifying therapy of a potentially devastating disease like Alzheimer's dementia, and we have a tool that is exquisitely sensitive to pick up changes in the brain that are relevant to that disease that may occur a decade or more before the manifestation of symptoms, and we can slow down or prevent that from happening. We're in a really